Welcome to this introduction video for the eDNA Citizen Science Sampling Kit. In this video, we will show you how to assemble the kit, collect a water sample, and preserve the environmental DNA for analysis. The standard items included in this kit are a dual filter capsule with connector, a 50 ml syringe, and a small pre-filled syringe containing preservation buffer. These items are included in every kit. Depending on the kit, additional items may be included, such as gloves or measurement strips. The first step is assembling the sampling kit. Remove the dual filter capsule from the packaging. Inside this package, you will also find a loose blue cap. Set this cap aside. You will need it later. Next, remove the 50 ml syringe from its packaging and connect it to the plastic connector on top of the dual filter capsule. Make sure the syringe is firmly attached. The syringe will only fit on one side of the connector. To start collecting a water sample, place the inlet of the dual filter capsule in the water. Pull back the plunger of the syringe to draw water through the dual filter capsule. The syringe will fill with water that has passed through the dual filter. To avoid drawing air into the filter, keep the inlet of the dual filter capsule submerged while drawing water. This is very important. Once the syringe is full, push the plunger to discard the water. If it's more convenient, you can lift the dual filter from the water during step 3, when you discard the water. As long as you keep the inlet of the dual filter capsule submerged when drawing in water during step 2. Repeat these steps approximately 15 times, or until the dual filter becomes clogged. Each cycle, or subsample, consists of drawing water through the dual filter and emptying the syringe. To get a more representative sample of the environment, try to collect water from multiple nearby locations. For example, filter one syringe full of water, discard it, and then move about 10 meters to repeat the process. Sampling along a small gradient like this helps capture environmental DNA that may be unevenly distributed in the water. Once sampling is complete, the dual filter capsule must be dried. Lift the dual filter capsule out of the water and pull back the syringe plunger as far as possible to draw air through the dual filter. This removes any remaining water inside the capsule. The next step is to preserve the eDNA captured on the dual filter. First, detach the large syringe from the inlet side of the dual filter by removing the silicone tube. Close this side of the dual filter by firmly attaching the loose blue cap that you placed aside earlier. Now turn the filter upside down so that the inlet side is facing up. Take the pre-filled small syringe, remove its blue cap, and connect it to the inlet of the dual filter. Slowly push the plunger to inject the preservation buffer into the filter capsule. Once all liquid has been injected, wait a few seconds and then slowly pull the plunger back to approximately 3.5 milliliters. This is an important step because it releases pressure inside the filter. Remove the syringe and place the blue cap from the small syringe onto the inlet of the dual filter. Finally, record any required information according to the provided instructions, such as location and date. Place all components back into the resealable bag and seal it properly. The sample is now ready to be sent off.